What if every gene on Earth could be mapped just like a city? Welcome back, everybody. I am Sayyada Atika, pursuing MSc Genetic Engineering from School of Biotechnology, DAVV, Indore. And glad to have you all here. Today, we are exploring something truly fascinating. In today's video, we'll explore how bioinformatics and geoinformatics connect data, life, and our planet in amazing ways. Let's jump straight into the world where biology meets technology. To start with, bioinformatics. What is bioinformatics? It is an interdisciplinary field that combines biology with computer science, mathematics, and statistics. It helps us manage and analyze huge biological databases. Things like, and now what includes the biological databases? Things like DNA, RNA, and protein sequences that are too complex for manual study. Data are raw material that we do have. Then we manage them, managing biodata, arranging them, then using it for problem solving, data analysis, and then discovering and development for human benefit, like drug discovery and health tools. Thanks to powerful computers and new tools, bioinformatics helps us decode life itself. Now, biological databases. A biological database is a collection of biological data stored digitally, so it can accessed, added, retrieved, or analyzed. These databases are managed by Database Management System or DBMS. Now, moving on, what are types of biological databases that are included in this? On the basis of nature of data, they are classified into three main types: biological databases on the basis of structure, sequence, and other. What includes the structure? Based on the structure of the biological data, we have classified it into PDB, SCOP, Keth. And on the basis of sequences, depending on if it's a protein sequence or if it's a nucleic acid sequence, we have divided further into two groups. Protein sequence databases includes SwissProt, PIR, Tremble, ProCite, PFAM, and nucleic acid databases, biological databases includes GenBank, DDBJ, EMBL. Don't worry, we'll go through all of these one by one in the upcoming slides. The other that are classified as Relibase, Rebase, and GPCRDB. Moving on, biological databases are also classified on the basis of their source, primary databases and secondary databases. Let's get delved into it. What are actually primary databases and secondary databases? So the primary databases are the original or raw experimental data like original DNA or protein sequences. And these are generally submitted by scientific community based on their experiments and discoveries. Now, what are secondary databases? Secondary databases contain information derived from primary databases, raw data that are derived from the primary database. For examples, patterns, motifs, or structures that are derived from the primary database of the sequence of either protein or nucleic acid. And then comes the composite databases. Composite databases are the combination of data from different sources and remove the duplicates, the non-redundant sequences. It includes the non-redundant sequences, that is, it does not repeat any data from primary or different databases. And it is also used for comparing the data. Next, we'll delve more into what are primary databases. Now, based on nucleic acid databases, protein sequence databases, and protein structure databases, there are different kinds of primary databases that are available nowadays. The very popular nucleic acid databases includes GenBank, EMBL, DDBJ. Protein sequence databases includes PIR, SwissProt, and UniProt. Protein structure databases includes PDB, and metabolic databases includes KEGG, KEGG. Nucleotide sequence databases. Of course, nucleotide, as the name suggests, it includes the sequence of nucleotides, nucleotide sequence entries, and there are data repositories that accept nucleic acid sequence and make it freely available to the public. The main highlight is that they are freely available to the public. Main includes GenBank, EMBL, and DDBJ. All three are members of International Nucleotide Sequence Database Consortium, INSDC, and interchange data. These three are the main source of primary nucleotide sequence database, where the data can be retrieved and even accepted. And these are the databases that can interchange their data for further studies. And all of these three main nucleotide sequence databases are the part of INSDC. Moving on to explore more about GenBank of NCBI, hosted by National Center of Biotechnology Information, that is NCBI, situated at the campus of US National Institute of Health, USA. GenBank stores publicly available nucleotide sequences and their annotations, and this is sourced from USA. It facilitates direct submission of sequence database and user-friendly. Direct submission and user-friendly. You can even get a unique accession number after quality checks. The link is provided here for the accession of the NCBI through internet. You can access it anytime through NCBI's website. Now let's explore its homepage. This is the homepage of NCBI, National Center for Biotechnology Information, and here it is mentioning GenBank. This is the drop down menu where you can select the nucleotide. Even you can insert any accession number over here and search for required nucleotide sequence as per your convenience. And this is a historical information given over here, and you can also get the latest notice of the site. Now, moving on, DDBJ, that is DNA Data Bank of Japan. Started in 1986, hosted now at National Institute of Genetics, Japan. This is the nucleotide sequence database of Japan, and it is mainly from the scientists of Japan, but it also accepts the data from researchers from all over the world. And this also shares its data from GenBank and EMBL. About 99% of nucleotide data in INSDC submitted by Japanese researchers through DDBJ. Okay, so you can see the importance of DDBJ as it contributes most of the INSDC data. Also provides tools. It also provides tools, accession numbers, and training programs for bioinformatics beginners. Right. Internationally recognized accession number. It gives the nucleotide sequence its unique accession number, and they can conduct many courses for the beginners to reduce the complexity of biological data. Again, you can access it through this website on the internet. Let's see its homepage. This is the DDBJ's homepage. It's based off Japan. Accession number, how to use, the availability of the data can be viewed over here. You can search through if you want to go particularly for a DNA or PDB, you want to see the structure of the DNA or RNA or any sequence. So this is the homepage of DDBJ. Now comes EMBL. EMBL, European Molecular Biology Laboratory Nucleotide Sequence. It was first established in 1974 and hosted at UK. So the European Molecular Biology Laboratory stores genetic sequences as they appear in nature without artificial modification. And it is a non-profit research institution supported by 20 European countries, as it is based in Europe and Australia for molecular biology research. Again, the data corresponds to wild type sequences without mutation, genetic manipulation. You can access this nucleotide database through this website as well. This is the EMBL homepage. You can go through the researches, services, trainings it provide, or if you want to search something very particular, you can go through the search button as well. It's very user-friendly. Now comes the protein sequence database. Apart from DNA, proteins are equally important, as you all know. Proteins are equally important as DNA. We have protein sequence databases like SwissProt, PIR, and UniProt, which provide detailed information about protein structure, function, and annotations. These are unique for protein sequences. Now, moving on for our first protein database, that is SwissProt. So SwissProt was developed by Swiss Institute of Bioinformatics, SIB, and European Bioinformatics Institute, EBI. With the collaboration of these two institutes, we got SwissProt. High quality, manually annotated protein sequences and created in 1986. It provides high level of annotations with functions of proteins and relevant information about particular protein. It has two parts, UniProt and UniProt KB Swiss and UniProt KB Tremble. And you can also access this through this website. This is the homepage of SwissProt. Data resources, computing, analyzing. You can search by database, search through the database by name, category. Very user-friendly and very easy to use. URL, full name, and the description is also given over here. You can search here as well. You can directly input your need into the database and it will provide you with the output. Moving on, PIR, Protein Information Resource Database, established in 1984 by National Biomedical Research Foundation, NBRF. It is an integrated public bioinformatics resource that supports genomic and proteomic research and scientific studies. Again, it is another protein sequence database. It is developed by NBRF and it helps in research and identification, also interpretation of protein sequence information. This is the website link. You can go through it. This is the PIR page. Again, about PIR resources, search analysis. You can get here a very precise and clean homepage without much of the data. Yeah, here you can input your accession number or ID for particular sequence of protein. UniProt. Again, UniProt is another kind of protein sequence database. It is very comprehensive, high quality, and freely accessible resource of protein sources. And it is it is the UniProt consortium comprises the European Bioinformatics Institute, the Swiss Institute of Bioinformatics, and the Protein Information Resources. It includes all these three institutes and becomes a comprehensive protein sequence database. This composed of 4 components, and all of them are for different uses. This is the homepage where you can see the 4 components: proteins, species, protein clusters, and sequence archive. This is very comprehensive and high quality database, and very widely used by researchers and innovators for the protein analysis and interpretation. Then comes the structural database, protein data bank. Protein data bank, now what comes the need of structural database? Many proteins which exhibit a common evolutionary origin can show different structural similarities or dissimilarities. So to understand their evolution relationship and their function, we need structural database so that we can see and analyze and interpret the relationship between their sequence and their function. Dissimilar proteins exhibit changes in primary, secondary, and tertiary quaternary structures. Basically, we see the structure of proteins in this database. Similar or dissimilar protein structures can be predicted. So it is very helpful in analyzing the similarities and dissimilarities of same sequence protein by looking at their structures, primary, secondary, tertiary, or quaternary structures. Comes the PDB, that is Protein Data Bank, stores 3D structures of proteins and nucleic acids. Understanding these shapes helps scientists study how molecule works and interact. This is very important for scientists to analyze a nucleic acid or protein's structure. And this is maintained by Research Collaboratory for Structural Bioinformatics, RCSB. This is Single Worldwide Archive of Structural Data. This is Single Worldwide Structural Database, PDB. Although it says Protein Data Bank, but it also contains nucleic acid structures too. And where are these obtained from? These are obtained all from the X-ray crystallography and NMR spectroscopy. The structures of nucleic acid and proteins are obtained from these 2 techniques and are stored in the Protein Data Bank, getting each structure a unique accession number and ID. This you can also access at the PDB. This is the homepage of PDB where you can analyze the 3D structure of proteins or nucleic acid. You can deposit, you can search, you can visualize, you can analyze. Very, very helpful for the beginner or a highly educated and innovative research field. Now comes, these are the primary databases that we have discussed that we get from the raw materials derived from the scientists and the experimental labs directly. Now now comes the secondary databases. What are secondary databases? It contains data derived from result of primary data. Okay, they are either created manually or generated automatically. Or why these are important? Because they give us information about mutations and evolutionary relationship. Some examples of secondary database are ProCite, PFAM, blocks, prints. So these databases, ProCite, PFAM, now these databases will use the data, raw and original data from primary databases like the one we discussed above, GenBank, PDB, or UniProt, SwissProt, NCBI, EMBL. These secondary databases derive the main raw material from the primary databases now for further studying of mutations or evolutionary relationships or any other research studies. ProCite. We'll go through some of the secondary databases. ProCite, protein database, consists of entries describing protein families. Now the sequences derived from the primary data, they create protein families, domains, functional sites, as well as amino acid patterns and profiles them. They arranges them in a proper manner. And who does all this? This database is curated by Swiss Institute of Bioinformatics, that is SIB, and tightly integrated into SwissProt protein annotation. So you can get from where it is coming. It is coming from the primary database SwissProt. We have already discussed about SwissProt. Now come PFAM. PFAM is again a protein database, protein family, multiple alignments generated and gives the hidden Markov models. It is enhanced in what ways? That it can also give multiple alignments and Markov models. The general purpose of PFAM database is to provide a complete and accurate classification of protein family and domains. This is its homepage, available at EMBL and EBI. That is, it is situated with European databases. It derives PFAM. PFAM derives its primary data from EMBL. I hope this is clear until now. Now, moving on to another topic, that is geoinformatics. Now, let's move from molecules to maps. Geoinformatics is a science and technology which develops and uses informations, which deals with collecting and analyzing and visualizing geographical data related to geography, geosciences, and related branches of engineering. By using the tools like satellites and GIS, that is geoinformatics software. The definition says art, science, or technology dealing with the acquisition, storage, processing, production, and presentation of geoinformation. Branches of geoinformatics. Branches of geoinformatics include remote science sensing, geographic information systems, cartography, global navigation satellite systems, and photogrammetry, and DBMS. These are its 6 branches of geoinformatics. We'll go one by one to each one of them. Remote sensing. Remote sensing is the means collecting information about Earth's surface without direct contact. Means we are collecting the Earth's information but without its direct contact. Then how are we doing without direct contact? We are doing it through satellites or aircraft sensors. It helps monitor land, forests, water bodies, and environmental changes. This is mainly used for analyzing or monitoring forests, water bodies, and environmental changes. How many types of remote sensing are there? There are 2 types, passive and active. As you can see here, passive instrument is there, and this in this active instrument there is a almost a direct visualization and monitoring. Now comes geographic information system, that is GIS. GIS is a computer-based information system that analyzes and visualizes the geospatial data. What does geospatial means? Data which can be represented using geographical coordinate system, means latitudes and longitude. I hope latitudes and longitudes are clear to you. Latitudes means these are longitudes on the Earth surface, and these are the latitudes. These are all imaginary lines on the Earth's surface for its better representation. It combines maps and data to study patterns. You can see, for example, mapping roads, rivers, soils, types, or urban growth. This is all the same thing I have already explained. Storage, retrieval for the geographic data, and it can include hardware, software, procedure, and appropriate GIS. Storage, retrieval, display are tools provided by GIS. Okay, well, GIS in general. What does it include? Mapping of roads, network settlements, water bodies, hospitals, railways, airports, soil types. These are all the. Now comes what are the components of GIS. Components of GIS includes hardware, software, data, people, and methods. These all includes in a GIS system. Now comes the cartography. What is cartography? It is the art of art or technology that deals with making of maps. While GPS, that is, then how it is different from GPS, that is the global positioning system. It is different. It uses satellites to pinpoint any location on Earth accurately, right? GPS system is used to pinpoint any location on Earth accurately, while this deals with the making of maps. Now comes another point into our geoinformatics, that is global navigation systems. What is global navigation systems? Global navigation systems includes GPS satellites, master stations, monitor stations, high accuracy positioning, and some satellites. These are interconnected to provide GPS or exact location of the required person or place. The global navigation system, positioning system, that is GPS, satellite based, it is satellite system, and that provides location and time of information in all weather. The main benefit of it is that it is available in all weather conditions or anywhere on or near the Earth surface. It is majorly dependent on the satellite system, where different stations, monitor station, master stations are also the part of it. Now comes the photogrammetry. What is photogrammetry? It uses aerial photos to measure distances and create accurate 3D maps. These are all the different types to use, the different types to generate geography of Earth. Photogrammetry uses aerial photos to make 3D structures, and it gives accurate 3D maps. That is very important technique for surveys and mapping. Now comes our DBMS, that is database management system. It is a software designed to assist in maintaining and utilizing large collection of data. Like in bioinformatics, geoinformatics also relies on database management system to store and manage large volume of spatial data. Okay, now we have collected all the geoinformatics data, the geographical data that we needed through photogrammetry or GPS or through cartography. Now where to store them? So we need DBMS for that, which uses the database like Oracle and SQL for the large collection of data. So in summary, bioinformatics organizes the data of life, while geoinformatics organizes the data of Earth. Together, they help scientists make smarter decisions, from disease mapping to environmental protection. Thank you for watching, and if you found this helpful, don't forget to like and subscribe for more science insights. Thank you so much.